Welcome to my teacher program. Today we are going to discuss an important topic which is very useful for grade 10, 11 and 12, and this is the basic phenomenon which is taking place in almost all living organisms. So I will show you a small video clip. You please identify the topic, then we will come back to that. You can see the bacteriophage attacking a bacteria and phage trying to penetrate into the cell. Countless living cells. The earth is teeming with countless living cells. You can see the different types of cells moving across. Fusion of two cells. We have 100 trillion cells in our body. I hope you will be able to identify the topic, today's topic, by seeing the structure of cells and number of cells. Now I will show you another process by which you can see here what is happening to the nucleus. Cells start migrating into various regions. The entire organism is being formed by different organ systems. So here you can see that cells do not live forever. Now let us say when cells do not live forever, we have seen that a human body or any living organism is made up of different types of cells and trillions of cells. So how do new cells form? Because you have seen that cells do not live forever. Now we have to discuss a process by which cells multiply or increase in number. This process is known as cell division. So today's topic is cell division. Cell division, it is for grade 11. Of course, it's there for even grade 10 students. Now let us discuss what are the different methods by which a cell divide. Before going to that, we should know that why should a cell divide. Cell has to divide. We have seen that to increase the number of cells in the body, because each cell has got a particular lifespan. So after achieving a particular lifespan, cells divide, and also cells divide increasing the size as well as the growth. So this type of cell division taking place both in animal as well as in plants. Both animal cells as well as the plant cells undergo cell division. Of course, in plant cells you might have studied there are different areas, regions or zones we can identify where the cell division takes place. Based on the property of cell division, plant cells are classified into meristematic cells, and permanent cells. Permanent cells. So what is the function of these meristematic cells? Meristematic cells are present on the meristems, or I have mentioned it helps for the cell division. So which helps to increase the size, or which helps in the growth of the plant. So we have seen that there are different types of meristematic cells which you might have studied in the anatomy chapter, plant anatomy. There are different types of plants meristematic tissues. Today we are discussing about the cell division, but let us see what are the meristematic tissues which you have studied. That is the apical meristem, then lateral meristem and intercalary meristems. Permanent cells cannot divide, which does another function. So now we are going back to cell division and let us discuss what are the two different types of cell division. There are mainly two different types of cell divisions, that is mitosis and meiosis. So, we have seen that each cell, after attaining a particular size, it undergoes cell division. So, each cell has got a cell cycle. So, let us see what is cell cycle first. Cell cycle. Cell cycle has various phases. These phases of cell cycle, for example, we can see that there is a phase which is known as interphase. Interphase. Interphase is a large phase of the cell cycle. Then comes to mitotic stage or mitosis. So, we can see that the cell cycle consists of an interphase and mitotic phase. Interphase is again divided into three different phases. These phases are G1 phase, then S phase and G2 phase. So, we have seen that the cell prepares itself for the cell division during the interphase. Then the cell division starts and the various steps taking place in mitotic cell division. Now, let us see the slide. I will show you the cell cycle. Here you can see the cell cycle. The first growth phase, you can see the growth and normal metabolic roles, that is in the G1 phase. So, G1 phase is the first growth phase. Growth and normal metabolic roles. The cell undergo the normal metabolic processes. Then comes to the second phase. The second phase is the synthesis phase, or it is also known as what? S phase. In this S phase, what is happening is DNA replication takes place. Normally, you are getting confused between this DNA replication and number of chromosomes increases, doubling of chromosomes. So, please do not get confused. In S phase, chromosome number remains same, and only what is multiplied, only the DNA content will be increased. So, DNA replication taking place, which helps to increase the number of DNA in the cell, but the chromosome number remains same. Then comes to the third phase. This phase is the G2 phase. G2 phase, that is the second growth phase. In this second growth phase, we can see that growth and preparation for mitosis taking place. So, these are the three phases which is coming under what? Interphase. Hope this part is clear to you. Interphase is the long phase which is having three stages: G1 phase, S phase and G2 phase. Now, we will move on to the mitotic phases. Now, let us see what are the different mitotic phases. As mentioned, mitosis has various steps. The first step in mitosis is prophase. Prophase, then comes to metaphase, anaphase, telophase. So, as we have seen that these stages taking place in the vegetative cells, because meiotic cell division taking place in the germ cells. So, mitosis takes place in the vegetative cells. It has four phases, and after these phases, what is the outcome? The outcome is one cell divides into two cells, and which is having equal number of chromosomes. Now, let us see what is happening in prophase. So, let us consider a cell, a normal cell which is having a nuclear membrane, and it is having nucleolus, and you can see the chromatin threads. So, these are the various stages we have seen that there is a nuclear membrane, nucleolus, then chromatin threads. Now in prophase, here we can see that condensation of chromatin materials taking place. That is, chromatin materials will get condensed and which attain a definite shape. We can see that chromosomes become more clear condensation taking place. Then in the late prophase, so normally students are asking up to which stage the prophase taking place. We cannot easily demarcate each phases because it's a continuous process. Still we say that once the nuclear membrane disappear, nucleolus disappear and chromosomes are clear, we say that that is the end of prophase. Now in metaphase we can see that, then also remember that the doubling of chromosomes taking place. Chromosome number will be doubled. So we can see that each chromosome, before it was having only a centromere and 2 chromatids. Now it has 4 chromatids. So we can see that the chromosome has 4 chromatids, that is on a centromere 4 chromatids are attached. So what are the changes taking place during prophase? Nuclear membrane disappear. Now we have to show that the disappearing of the nuclear membrane, then we can see that nucleolus disappear, then we can say that doubling of chromosomes start doubling. Then this stage, after this stage, cell will undergo the next stage. This stage is known as metaphase. So what is happening in metaphase? Now let's see, in metaphase we can see that these chromosomes will arrange in an equatorial plane. So if you consider this, we can say that these are the 2 poles of the cell and in the equatorial plane. So chromosomes will arrange on the equatorial plane and spindle formation taking place. So from where do the spindle fibers arise? Spindle fibers forms from the centriole of the cell. So that we can see that even in the last prophase, we can see the centriole divides and moves on the 2 poles of the cell. So 2 centrioles will be there. These centrioles will move on the poles of the cell, as I said. Then we can say that the chromosomes arrange on the equatorial plane. Equatorial plane. Now we can see that the spindle fibers, what is spindle fibers made up of? These are made up of microtubules, and these spindle fibers attach on the centromere of the chromosomes. Spindle fibers attach on the centromere of the chromosome. So here we can see that during metaphase is the easily identifiable phase, and normally we use this metaphase for all the karyotyping procedure, because during karyotyping we have to identify the chromosomes based on the position of the centromere, also based on the shape as well as the size of the chromosomes. Now these chromosomes are better studied at metaphase of the cell division, because it is more visible in metaphase. Because cells will be arrested in metaphase using some chemical substances like colchicine, and the chromosomes will be studied during metaphase. Now let us continue this. We can see that during metaphase the chromosomes arrange on the equatorial plane and the spindle formation taking place, and later what is happening is the centromere breaks. So here you have to be very careful. You should know that what is the difference between the anaphase 1 of meiotic cell division and anaphase of mitosis. So meiosis we will discuss later after this. Now please understand how does the anaphase taking place. During anaphase, centromere breaks and chromatids get separated. Here you can see that it start moving to the poles. Okay, so we can see that this centromere breaks and the chromatids are pulled. Chromatids are pulled into the two poles of the cell. This phase is the which phase? Anaphase. Now this is the metaphase and this is anaphase. Hope this is clear to you. Now what is happening is we can see the last phase, that is the telophase. Again we can see that these chromosomes moves on the corner, that is into the poles, then into the poles, again chromatins, nuclear membrane, nucleolus forms, or nucleolus, nuclear membrane, and chromatin materials will be formed. So here we have seen that a cell has got two nucleus. This process of mitosis is known as karyokinesis. Karyokinesis means breaking of nucleus. Now let us see what happens to the cytoplasm. The cytoplasm of the cell, in the case of plant cells, a cell wall or the middle lamella, we can see that a plate formation taking place. Cell plate formation. This cell plate formation takes place and cell plate becomes the middle lamella of the cell wall. You might have studied the structure of a cell wall. Inside the cell wall there is middle lamella, calcium pectide. So we have seen that this middle lamella is formed from the cell plate. So as a result a cell will be divided into two cells. Now in case of animal cells, cell plate formation won't take place. Instead what is happening here in animal cells, a cleavage furrow will be formed. A cleavage furrow will be formed, and this cleavage furrow will move inward movement of the cleavage furrow of the cell membrane, and as a result a cell will be divided into two cells. Now we understood the difference in cytokinesis in plant cells as well as in animal cells. One more you have to remember, when spindle formation takes place it is from the centriole, as I said, but you have studied that centrioles are absent in plant cells. So from where do the spindle fibers arise? The spindle fibers arise by the joining of microtubules. So remember that cilia, flagellas, then centrioles, these all are made up of microtubules. These are the particular cytoskeleton which is present inside the cell. Now let's see the various stages once again on the slide. Here you can see the interphase is given. In the interphase centrioles are shown. Centrioles, then chromatin, then comes to the next phase, that is the prophase. What is happening here in the prophase? You can see the doubling of the chromosomes, nucleolus, then sister chromatids. Next phase, that is the metaphase, where chromosomes are arranged on the equatorial plane, spindle formation, then comes to anaphase. In this anaphase you can see the chromatids are separated and they start moving to the poles of the cell. A cleavage furrow is formed. Since this diagram representing the animal cell, you can see a cleavage furrow is formed and two nuclei is formed, and after the karyokinesis the process of cytokinesis proceeds. Now ultimately we can see that a cell becomes two daughter cells. Now what happens to these daughter cells? These daughter cells will undergo the first step, that is the G1 phase of metabolic reactions. Now we can see the G1 phase or the normal metabolic reactions taking place inside the cell, then comes to the DNA replication, then G2 phase, then comes to mitotic phases. So hope this is clear to you, the cell division, and as I have mentioned, the cell division process as well as the diagram you should know very clearly, because this helps you to learn more about the chromosome structure as well as the process which is taking place inside the cell. Then also you can understand how do cells divide and increase in number, or the growth of the organism taking place. So now we will see the next another type of cell division, meiotic cell division, after a short break.